we have already read about the rods and cones, and the rods and cones consist of the chemical sensitive chemicals, light sensitive chemicals. In case of rods, that is known as rhodopsin, and the light sensitive chemicals that is present in the cones are these cone pigments or the color pigments. Okay, there are three types of pigments there. Now, while we are discussing on this photo transduction, will basically focus mainly on the rhodopsin, but similar type of mechanism is also occurring on this pigment that is present in the cone. So for the easiness, where in the books, in different books, you could see that the rhodopsin, okay, they are talking about mainly on the rhodopsin. Clear?
Yes, sir.
Now, if you look at the visual pigments and decomposition by light, okay, what is that? But decomposition is not permanent, that is temporary, transient type, okay? Decomposition is transient type, that we will discuss on more detail. If you look at here, visual pigments like rhodopsin is composed of the protein. What is that protein? I have already told that there are visual pigments are present in the rods as well as in the cones. And the rods, when they are there, they are known as the rhodopsin, and they are consist of the protein known as scopopsin, and there are also the carotenoid pigments, retinol, they are also known as the retinine. Okay, so if you combine this retinine plus scopopsin, then it forms the rhodopsin, that is rod pigment. So these two get combines scopopsin and the retinine, or you can tell it as a retinol. Okay, retinol, that is forms the rhodopsin. So cis form, there are also cis and trans form of retinol is there, okay, or retinine. So cis and trans form is there. So only this cis form of the retinol can bind with the scotopsin, and they will form the rhodopsin. So when the light strikes, okay, when the light strikes, then this cis form will be converted into trans. Okay, this cis form of the retinol converted into the trans, and during that process, there is separation of this retinol from the scotopsin, and that is known as bleaching. That is also known as bleaching. That I will show you in the figure as well. What is that? What is bleaching? But you should be knowing that right now. You should be knowing that the cis form of retinol will combine with the rod of scotopsin, and they will form the rhodopsin. Clear? Am I clear?
Yes, sir.
Now let me discuss on this part. Okay, if you look at here, the rhodopsin, light pigment and decomposition by lights. Okay, and this also shows there is a synthesis is also going on. So rhodopsin, when the light strikes on these visual pigments, okay, in case of the rods, by this photoreceptor, then that will convert it on this rhodopsin, and then like this, okay, various steps are there. So this partly split combination of all trans-retinol and scotopsin is called rhodopsin. So there is partial split is there. Okay, there's this cis. I have already told you cis retinol is there, plus cis retinol plus scotopsin is forming the rhodopsin. So when the light strikes on that, then at a few nanosecond, within a nanosecond, then it leads to formation of this structure. Okay, and this partial split combination of trans-retinol and scotopsin is known as rhodopsin. Within this millisecond or microsecond, they will ultimately within second they will convert it to the. Look at it here, then that they will convert it to the all trans-retinol, isn't it? All trans-retinol. So cis form of this retinol will ultimately converting into the trans-retinol. Okay, and if you look at here, this one, this meta-rhodopsin second is called the activated rhodopsin. Okay, this is the activated one. This one is activated rhodopsin. And cis, you can see the short, this is trans. Actually, it can go to the isomerization. The enzyme is there already. Then it ultimately can reach the cis retinol, and this will be there. And this is photo can combine with cis, and they will ultimately form the rhodopsin. Am I clear? This mechanism is going on. So if the question is asked about visual pigment and decomposition and composition, this is the transient decomposition. This is transient one, and after that, certain period of times, they will also be sensitized. Synthesis is going on. Now let me discuss on. Okay, this I have already told you that when light is absorbed by rhodopsin, isomerization occurs in visual pigment. What is that isomerization means? Cis cis form, cis rhodopsin will be converted to the all transretinal. This is known as isomerization. Okay, so when the light is absorbed by rhodopsin, isomerization occurs in visual pigment, that is cis form change to all transretinal form. And when this isomerization is going on, this isomerization ultimately leads to the separation of the retinal from the opsin. That is also known as bleaching. If you look in the further in this diagram, also you can see there is bleaching is occurring. Isn't it? This opsin, scotopsin, and this structure is being separated, and this separated structure there is known as bleaching. What if the question is bleaching? Then you should be telling that there is separation, okay, of this retinal from this opsin or scotopsin when the light strike on there. It is due to the cis component of the retinal change into the retinal. Okay, all into the all trans retinal. Am I clear? Am I clear? Please let me know.
Yes, sir.
Now, if you look at here, now this is the photo transduction. Okay, this is the mechanism. Just I told you the mechanism related to the decomposition of this retinol and slightly on how they are being that will be reformed, isn't it? This is the transient mechanism was there, composition mechanism was there. Now, if you look at here, this is the photo transduction means what is that? Photo means light, isn't it? Transduction means the what is happening within the photoreceptors. Okay, it means one form of the energy will change into the another form of the energy. Okay, so this is known as phototransduction. Means light is converting the one form of information to the another. So due to the light, that was hello. So that will strike on the photoreceptors. Here in this case, this is the rhodopsin. So this is known as phototransduction. So light will strike on the these photoreceptors. In this case, the figure is shown for the for the rhodopsin. So what is the mechanism that is going on? What's one form of energy is being converted to another form of the energy? How the information is going on? Okay, it's just like that. If you look at here, this is the one of the segment. Okay, if you look at here, this is the you have already read maybe that in the rods there are the structures are there, outer segment, inner segment is there, isn't it? Have you read that? Inner segment, outer segment. Deepika, in the previous class, have you read that these photoreceptors having outer segment, inner segment, and these two segments are being connected by another structure? You have read, isn't it?
Yes, sir.
So if this mechanism is occurring in the outer segment, okay, this is occurring in the outer segment. What is there? If you look at here, visual pigments, that is rhodopsin. Okay, this is the rhodopsin. Now, when the light strikes on this these pigments, then you can see that this is the opsin. Okay, this blue, this one is the blue, this one is the opsin, scotopsin, and this one, okay, this one is the retinal, and this together is known as the rhodopsin. If you look at here, when the light strikes, then this angulated structure, this is angulated structure of this cis retinal, okay, converted into the ultimately to the trans retinal, and it is straight one. So when there is this mechanism going on, that will cause the activation of this structure, that is the transduction. So they will activate the transduction, and that activated transduction further activates the enzyme, that is a phosphodiesterase. Okay, this is the enzyme, look at here, phosphodiesterase enzyme, and that phosphodiesterase enzyme, when get activated, they will convert CGMP into the this GMP. So CGMP converted GMP. Okay, this is by the enzyme phosphodiesterase, which is being also activated by transduction. The transduction, this transduction, I have told you, this is being activated when the light strikes on the these visual pigments and due to the conversion, and that within conversion of cis to the transretinal. Okay, am I clear? Shall I repeat it again?
Actually, repeat garden, sir.
Okay, if you look at here, that I have told you that this one is here and this is cis retinal. This whole structure, okay, this whole structure is the rhodopsin, isn't it? So rhodopsin is there when the light strikes, and if the light strikes, then this cis retinal 11, okay, 11 cis retinal, this angulated, look at this angulated form is there. Now that is converted into straight one, that is the transretinal. Isn't it? I have already told you different steps that is transretinal. Then within this second, they will convert into all transretinal. Okay, this has been not shown over here, but I just shown before, isn't it? So when this all transretinal is being formed, the bleaching is going on, isn't it? Bleaching means the separation of this retinal from the opposite scotopsin is going on. During that process, they will convert or they will activate this structure. This small structure is known as transduction, and this transduction activates this enzyme phosphodiesterase. When this phosphodiesterase enzyme get activated, then they will convert CGMP into the GMP. Okay, up to this it is clear.
Yes, sir.
Now, if you look at it further, in the resting, okay, when the light is not there, what is happening there? There is CGMP gated sodium channel, okay? So CGMP gated sodium channel was there, means CGMP was there, and it was, so CGMP was there to get this sodium channel. So when there is activated phosphodiesterase enzyme is there due to light. I have told you CGMP is converted into GMP. So when CGMP is not there, what will happen to this channel? This will get closed, isn't it? So no influx of the sodium is going on. Okay, so the level of the CGMP will be decreased due to the light. Ultimately, due to the light, isn't it? Due to the action of this phosphodiesterase on the CGMP. So CGMP gated channel, the sodium channel will get closed. So this get closed, so there is no influx of the sodium is going on. Okay, basically sodium is going. So most of the author is writing about the sodium. So sodium. Okay, am I clear?
Yes, sir.
So what is the ultimate mechanism? I have told you this thing is happening on, isn't it? Then what is the ultimate result because of this phototransduction that I have discussed again? Okay, if you look at here, this photoreceptor hyperpolarizes in response to light. But how? It is, now if you look at here, in resting condition, if you look at here, in resting condition, what is happening there? There is influx of the sodium is going on, isn't it? When the light was not there, so when the light was there, it closes the sodium channel. CGMP get a sodium channel. When the sodium is not there, means the cell is not being depolarized, isn't it? The cell is not getting the positive ions, isn't it? So that is the one thing. Another thing is that when the rhodopsin decomposes, decreases the conduction for the sodium ion in outer segment of the rods. Okay, this is the one example. Another is that this is thing that did you understand this one? When the rod of decompose, it decreases this rod membrane conduction for sodium ions in the outer segment of the rods. Means this all this phenomenon that I have just showed you, it is occurring in the outer segment of the rod, isn't it? So when the light is there, then it will closes this CGMP gated channel. Means there is no influx of the sodium. This is the same thing, isn't it? So when a rod of decomposes due to light, it decreases this rod membrane conductance for the sodium ions in the outer segment of the rod, isn't it? Is this clear?
Yes, sir.
Now another is that now the membrane goes on hyperpolarization. Okay, the membrane this membrane goes on hyperpolarization, the Dix membrane. Okay, now how it is going on hyperpolarization? So effect of light on photoreceptors. Okay, what is that? Stimuli causing the hyperpolarization. We have read that the stimuli in most of the cases the stimuli causes a depolarization, isn't it? When you have the action potential you have read, when the stimuli is there, then it causes the depolarization and the cell become active and the cell become activated, isn't it? You have read about for the cell or the electrically activated cell or excitable cell to get activated, the stimuli causes depolarization of cell, isn't it? Neurons and muscles, isn't it? Have you read that or not?
Yes, sir.
Now, here in this case, stimuli causing the hyperpolarization means stimuli is that it causing hyperpolarizing and it excites the rods. So this is the exceptional or not? This is the exceptional cases or not? The stimuli causing the hyperpolarization and it is causing the activation of the cells. Okay, how? Look at here. One of the example is closure of CGMP gated sodium channel, isn't it? This I have already told you when the light strike, ultimately there is closure of CGMP gated sodium channel is there. Another is sodium ion continue to be pumped out through the membrane of the inner segment. Okay, what is that? We haven't read that one. That I'll show you in the next slide. What is that? Okay, and so diminution of sodium ion flow is what excites the rods. Now, if you look at here again, okay, if you look at here again, then I have told you that isomerization is going on, and cis is converted into class, and that decreases CGMP, and this CGMP closes gated sodium channel get closes, so influx of sodium get decreases, and the cell get hyperpolarized. One of the phenomena is that another contributing factors for the hyperpolarization of the this rod rod is also in this one that I'll show you in the next slide. So the release of, if you look at it, the release of glutamate, okay, the release of glutamate that will decrease. Okay, in the resting condition there is, or in the dark the glutamate secretion is high, but due to this light, the release of glutamate secretion is low, and that low secretion of glutamate in turn activates the bipolar cell. We have read, isn't it? The photoreceptor cell. Photoreceptor cell is giving information to the bipolar cell. The bipolar cell is giving information to the ganglionic cell, isn't it? You have read that or not? You have read, isn't it?
Yes, sir.
So if you look at here, this is the thing that you should be knowing. And if you look at here, again, if you look at here, okay, if this one, okay, this dark current. So now, in absence of light, what is happening here? Just discuss you about what will happen when there is light, isn't it? Now, in the absence of light, what is happening there? Now, if you look at here, in the absence of light, there is dark current. It is responsible for the release of more neurotransmitter from the photoreceptors. Okay, more and more neurotransmitters are released from these photoreceptors when the light is not striking on these photoreceptors. If you look at here, there is this is outer segment, this is inner segment, this is outer segment, this is inner segment, and the outer segment there is passive influx of the sodium is going on, and this inner segment there is sodium potassium ATPase is there, that is constantly uh, this throw 3 sodium out and 2 potassium in. So there is the current is going on. This is known as dark current. Since this phenomena is occurring in the dark, so this flow of this sodium, okay, the flow of the sodium current is known as the dark current, and that causes the depolarization of the these cells, and that depolarization of the cell causes the release of the glutamate. But however, in the dark, okay, there is maximum release of the glutamate. However, I told you that where the light strikes on, there will be closure of this CGMP gated sodium channel that I have showed in the previous class, the previous slide. And another is that this one, okay, this sodium ion continue to be pumped outside through the membrane of the inner segment. Now look at here. Now look at here. So I told you the light will have impact on this, isn't it? Outer segment. This segment, isn't it? Light is having impact on this because there are the this rhodopsins are there, isn't it? Pigments are there, but this is not having impact on this, isn't it? So there is the closure of this when the light strikes. There is the closure of this CGMP gated sodium channel, and another is that this mechanism is also going on, isn't it? So this mechanism is also going on. So there will be the net loss of the sodium is also there, isn't it? So together, what will happen there? Together, from outer segment and inner segment, together they will cause the hyperpolarization of the these photoreceptors. Am I clear?
Yes, sir.
Is it clear or not? 5 of you, please let me know whether it is clear or not. Now, if you look at here as well, if you look at here, when the light strikes on this, okay, light strikes on this, hyperpolarization goes on, and the release of glutamate is less, okay. What in dark, look at here, the cell is on depolarization state, but the release of glutamate is high, and actually this is the cell is not activated, but this one the cell is being activated, okay. Hyperpolarization is activating the cell, and that activation leads to the decreased release of the neurotransmitter, that is glutamate, from the photoreceptors. Am I clear? Please tell me, 5 of you. If it is not clear, then it will be very difficult for you. Please let me know. 5.
Yes, sir.
1,
Yes, sir.
2,
Yes, sir.
3. 3. No one is there. It's not clear.
Yes, sir.
Okay, then for 5. 5 is not there. Now I'll see you. I'll call your name. It is not clear. Kafle. Rajul Kafle. Hello, Rajul. This night blindness. Look at here. This vitamin A is responsible for the formation of this ARSS retinal. So when this severe vitamin deficiency there, it leads to night blindness. Clear? We are not. People are not responding. So now visual pathway, that I will discuss on visual pathway. Now if you look at here, this I have already discussed in your practical classes as well. There about the visual pathway. Now if you look at here also, this okay, this retina, okay, there is retina is there. From the retina the information is going to the optic knob. This optic knob, what is that? Optic knob is actually the axons of the ganglionic cell, okay, the axon of the ganglionic cell, okay. This is most important, remember that, okay. this axon of the ganglionic cell is known as the optic knob. Okay, from the optic knob, look at here, the information is going to the optic chiasma, then from the optic chiasma, the optic tract, then the information is going to the lateral geniculate body, then from this optic radiation, the information finally reaches to the visual cortex. Okay, this and some of the information, if you look at here, is also going to superior colliculus. Okay, what is that? Okay, or the importance of that I will also discuss. Now, if you look at here again in the visual field, what will happen? This is the visual pathway. Okay, this is the visual pathway, and what will happen? What the impact of lesion? What will happen? What are the different types of defects on the visual field that will happen? I have already told you about the visual field also. So if you look at here, this is the visual field for this eye. This is the visual field for this eye. Now if you look at here, then this is the nasal side. Okay, this is the temporal side. This is the nasal side. The temporal side. And for this visual field, now if you look at here, the information is taken from the this temporal side. Information is coming to the nasal side. So this is the going on. Now if you look at here, then these are the photoreceptor cells are here. Photoreceptor cells and this ganglionic cell. This is the ganglionic cell that I have already told you. These form the axons. Okay, this axon is known as this axon is known as optic knob. Okay, this optic knob of the ganglionic cell. This is the optic knob. If you look at here from here, the optic knob is going like this, but here information is there and it's crossed over here this and they will then the information is going to another lateral geniculate body, similarly for this eye also. Now if there is lesion, okay, if there is lesion on this part, okay, that is known as blindness will occur in that eye. So this is the blindness is occurring on that eye. Now if the lesion is occurring at this level, okay, this optic chiasma, then this is not heteronomous hemianopia. Okay, then if you look at here, the lesion is occurring in this side, then this homozygous hemianopia. If there is lesion occurring in this here, again the same, okay, same time homonymous hemianopia, but macular sparing. Look at here, the sparing is going on, isn't it? This is known as sparing. Look at here, this sparing. This sparing is going on. Why this part is being not affected by the lesion present over here? It is because look at here, some of the fiber from lateral geniculate body. Okay, from here this will go to from the temporal lobe. This they will form the Mears loop. Okay, from here the information is going from this temporal lobe and they are coming here. Just this is the pathway. Now look at the press. This is there and this is being not shown over here. So they are the Mears path like this. And there is Mears path is there from the temporal lobe. They are going and they are joining here. That's why the macular sparing is there. Means there is these fibers get spared, isn't it? They will not be affected. Mears fibers will not be Mears loop or Mears fibers are not affected. That's why macular sparing will be there. Am I clear?
Yes, sir.
Now, I have already discussed why this phenomena is going on, isn't it? Why there is loss of this field, loss of field like this, isn't it? If you press the part, I have showed you in the practical classes as well, isn't it? That it is clear or not why there is reason, why there is defect on here and here, why there is defect over here and here, isn't it? I have made the this cracks and I have showed it also, isn't it? Now let me discuss. Okay, just I have also showed you tubular field of vision also, isn't it? Tubular field of vision, that I have also told you what type of tubular field of vision will be there. If you close your eyes like that, okay, if you now you can see. Now I will show you my face. Okay, please, can you see the video? No, to share your video. You have to. It's not sharing. I have just showed you that also, isn't it? Your practical class, isn't it? What type of vision will be there? What type of field of vision will be there? Deepika, भनेको थिएँ मैले, have I told you in your practical class or not?
भन्नुभएको थियो सर।
Okay, then let me discuss. Okay, again, if you look at it, the same thing is there. If you look at here, this input, now this one important. Okay, this information is there. Like same thing is there. Now look at this. What is that? Okay, the same thing. Look at heterozygous means opposite side of the visual field. That is heterozygous. Okay, hemianopia means half blindness. Macro sparing, white and macro sparing. Now again, if you look at here, now I want to show you this again. This figure might be it has been shown to you. There is photoreceptor rods and cones are there, and they are giving information to this bipolar cell, and from the bipolar cell, the information is going to the ganglionic cell, and this axon of the ganglionic cell, the axon of ganglionic cell, actually forming the optic knob, isn't it? They are forming the optic knob, and in between them, there is also the amacrines and horizontal cell are there. These amacrine cells and horizontal cell, they will modify the information that is coming from this these rods and cones. Then for the this amacrine cell modify the information that is coming from the bipolar cell, isn't it? Like that, okay? I want to show this figure now. Look at here, this axon of the ganglionic cell. Okay, this axon of the ganglionic cell. So ganglionic cell or I will be telling like different types of pathways. Now look at here. Now okay, there's large ganglionic cell. Can you see this slide or it is blurred? It is somehow blurred or you can see it.
We can see sir.
Okay.
अलिअलि thoughts.
और देखियो
देखियो सर।
Sorry for that. Now, if you look at here, this is the visual pathway that I have told you that from the retina, the information is going to the different structure and lateral geniculate body in the visual cortex, isn't it? But here in the lateral geniculate body also, there is some spatial orientation, representation of retina is going on. Okay, spatial representation of the retina is going on. Okay, which fiber will terminate on which layer of the lateral geniculate body. So there is some mechanism is going on there. So lateral geniculate body. This is lateral geniculate body. This is present in the thalamus itself. Okay, thalamus. This is the relay station. Thalamus nucleus. One of the nucleus of the thalamus is lateral geniculate body, and they have got 6 layers. 1, 2, 3, 4, 5, 6. Okay. So if you look at a large ganglionic cell, okay, ganglionic cell means these ganglionic cells, okay, the large ganglionic cell. I have already told you that this is okay. This is the ganglionic cell. This is the ganglionic cell. This is the ganglionic cell. Okay, this is the ganglionic cell. Okay. Now, so this large ganglionic cell is also known as M cell or Magnesium cell. So they are mainly concerned with movement and stereopsis. So they will add the information coming from the different types of the cones. And in case of small ganglionic cells, this again is ganglionic cell, small ganglionic cell. Okay, also known as parvo or P cells. They are mainly concerned with the this color vision. Okay, they are mainly concerned with color vision. So M ganglionic cells project to the magnocellular portion. Okay, magnocellular portion of the lateral geniculate body. Right now you may get confused what is the magnocellular and the parvocellular portion. Okay, this magnocellular and parvocellular are present. This is the small and large cell that is present in the lateral geniculate body. So this M ganglionic cell, this M ganglionic cells project means their fiber, their nerve fiber. Okay, the fiber projects to the magnocellular portion. So magnocellular portion, whereas this P ganglionic cell project to the parvocellular portion of the lateral geniculate body. Now, what is that magnocellular pathway? Okay, what is that magnocellular or parvocellular pathway means? From layer 1 and 2 of the layer 1 and 2. Okay, these are the this is the if you look at here, this is the magnocellular pathway. This is the magnocellular pathway. Okay, cells are large, but maybe you can see the layer maybe you can see this is small, but the magnum is larger. Okay, if you look at here, then from this layer 1 and 2, from layer 1 and 2, again if you look at from layer 1 and 2, they get combined and they will form the magnocellular pathway and rely on this visual cortex. Again, if you look at here, the parvocellular pathway, this is the parvocellular pathway from the layer 3, 4, 5 and 6. Okay, and they are terminating again on the visual cortex. If you look at here, magnocellular pathway, they will end on the this layer 4 of the foresight. But also if you look at here, the layer of the layer 4 is same thing, isn't it? So they are terminating on that. But main importance of parvocellular pathway, magnocellular pathway, that is coming from the lateral geniculate body. Okay, from this magnocellular and the parvocellular. Okay, uh, the again if you look at here, there is P fiber, isn't it? This large ganglionic cells and the small ganglionic cells. Okay, they are coming from here. Okay, they are coming from here, and they will, depending upon that, they will be terminating on that this lateral geniculate body. Main importance for this pathway. Okay, the question is asked like that. Magnocellular pathway mainly concerned with the this movement, depth and flicker. Okay, but this parvocellular mainly concerned with the color vision, texture, shape and fine details. Okay, so this pathway is responsible for this type of the vision, and this one is for this type of the vision. Am I clear?
Yes, sir.
Now, if you look at here, there is special P pathway from the interlaminar region of lateral genicular body to the bulb, this collar bulb. What is that? Special P pathway. Okay, this is interlaminar region. Okay, region. This interlaminar. This laminar like structure is there, isn't it? This 1, 2, 3, 4, 5, 6, isn't it? So in between that also, these interlaminar regions of the lateral geniculate body, some of the cells are also there. The dendrites of interlaminar cells means this cell. Okay, here the cells are there in the interlaminar region. So the dendrites penetrates the parvocellular pathways of the parvocellular pathway of the lateral geniculate body. Here, this one parvocellular, this one. Okay, here the cells present special type of cell. Here of special type of cell, special type of the cell. They will penetrate. Okay, and this type of the layer, and they will form another projection to the collar bulb. Okay, collar bulb that is present on the visual cortex, that is present on the, so there is input from the P ganglionic cell. Okay, you might get confusion, but you should be knowing that there are special P types of cells there, that is from interlaminar regions of lateral geniculate body to the collar bulb of the visual cortex, that is responsible for the perception or the concern with the color vision. Okay, and the bulbs, the color bulbs are also, there are different layers there, but mainly they will terminate on the layer number 2 and 3. Special type of P cell that will terminate on the layer 2 and 3 of the color bulb, and they are mainly concerned with the color perception. So, what if the question is being asked, which pathway is responsible for color vision? Then you should be writing that this one, the parvocellular pathway. Again, the question is being asked, which part of the cortex the color vision perception is going on this color bulb. Okay, you should be telling on that. Okay, there is special piece pathway is also there, that is also concerned with the color vision. Am I clear?
Yes, sir.
Now, if you look at here, again, I have just told you that ultimately the information is relayed on this primary visual cortex, isn't it? So, primary visual cortex, the information is relayed on both the magnocellular and parvocellular. Parvocellular mainly for the this color vision, and this magnocellular pathway mainly for the movement, depth, and flicker, isn't it? So, if you look at here, at this level, at this level, this B1 is known as the primary visual cortex. Okay, primary visual cortex, and most of the information will go to the primary visual cortex, and from there, even some of the information is also going to the other main areas. Okay, these are the different areas that is present in the visual cortex. Now, if you look at here, this B8, okay, this B8 is there. B8 is there. They are mainly concerned with the color vision. Okay, they are mainly concerned with the color vision. This is the most important. Okay, or this our vision. And if there is no which of the layer is responsible for detection of color, if the question is asked in B1, B2, B3, B4, B8 like that, that will be B8. Okay. So, and one book is writing this one, B8. Another book, just I have told you that they are mentioning about the color bulbs. Okay, they are also concerned with the color bulbs. Okay, they are responsible for the detection of the colors. Am I clear?
Yes, sir.
Now let me discuss on this near response and accommodation. Just I have already discussed, isn't it? They are increasing anti-curvature of lens. This pupillary constriction and convergence eyeball is going on. Now this I have showed you in the lab, and now I am going to show you the pathway. If you look at here, this is the pathway. Some of you have drawn it, but some of you haven't. So I suggest you to draw this figure. Now when I go finish this, please draw this figure as well. Now if you look at here, now I'll press on. Okay, now follow my this pointer. If you look at here, this is the light. This is the retina. From the retina, the information. If you look at here, the information from here, it is going to this area. I have already told you this lateral geniculate body that is present in the uh, thalamus. From here, the information is going to this visual cortex. This is the already I have told you this is the optic radiation, and from here now look at here. Up to this is the visual cortex. So we can see. But for the near response of accumulation, now the information has go from here visual cortex to the frontal eye field. Okay, frontal eye field. Information going to frontal eye field. From there, frontal eye field, the information is also going to this nucleus. One nucleus is parasympathetic nucleus. This parasympathetic nucleus. Another is the oculomotor nucleus. Okay, this is the parasympathetic component of oculomotor knob, and this is the this one is the motor third cranial knob. Okay, and from here, if you look at here from the parasympathetic division, if you look at here, then from here ciliary, from the short ciliary ganglia through ciliary ganglia, short ciliary knob, then it is going to the constriction of pupil. Okay, that the constriction of pupil will be going on for from here. Look at here the information from here. Okay, this is motor knob. This is innervating the medial rectus. So this will cause the convergence of the eyeball. Okay, am I clear? This pathway, this pathway clear to you? You should be able to draw this figure. Okay, this pathway I have similarly here also same thing is going on, isn't it? Am I clear?
Sir, actually, bonding fairy.
Okay. Correct, okay, if you look at here, these are the two eyeballs are there. Two eyeballs are there. So when I asked you to look far, isn't it? Far to the near, then what is going on? That is the visual reflex. Then that is known as the near response or accommodation reflex. When you see from far to the near, then what is happening there? When you see the near information from this retina, from this from the retina, this is going through this pathway, this optic knob, then optic chiasma, isn't it? Then from here, the information going to the optic tract and this lateral genicular body, and then this is the optic radiation, isn't it? The information we can see it, isn't it? Visual cortex, and even from the visual cortex, the information is also going to the frontal eye field. Okay, this is frontal eye field. This is frontal eye field. This is present in the this prefrontal cortex. I have already told you about the when I discussed on different lobes of the cortex, isn't it? So this frontal eye field that is present on the frontal cortex. Then from the frontal cortex, from two sides, if you look at it, the information is going to the two nucleus of the third cranial nerve. One is motor, another is parasympathetic. Parasympathetic division. This is again this here. This is the parasympathetic long ganglionic, the long fiber. This is short fiber, and this constriction of people going on. Another is that there is motor fiber. There is again they call the contraction of medial rectus motor. This also causes the contraction radial medial rectus motor muscle. So there is the convergence of the eyeball and constriction of the pupil. Isn't it? Am I clear?
Yes, sir.
Now, if you look at here, also I have told you that there is also the there is anterior curvature of lens is also increased. How that is going on? This is the explanation. If you look at here, it is not shown, isn't it? What this is the nucleus. This one the Edinger West Pole nucleus. Okay, this is the Edinger West Pole nucleus. This parasympathetic nucleus. This is in the West Pole nucleus for the contraction of the ciliary muscle. This is the ciliary muscle. Can you see? See the ciliary muscle. When you look for far distance, the ciliary muscle get relaxed. But when you see the near object, then the ciliary muscle get contracted. So this will be contracting, okay, and that will cause the increase in the anterior curvature of the lens. So lens become thick. Am I clear?
Yes, sir.
So if you look at this is the again this is the you should be knowing that, okay, in the West Pole nucleus. What is the in the West Pole nucleus means? Okay, this is the in the West Pole nucleus. Parasympathetic nucleus of oculomotor nerve. Okay, this one is in the West Pole nucleus, and this is the somatic nerve. Okay, means this is the motor nerve nucleus. Same thing. That is construction of people is going on. From this is the pathway this is being shown over here. Some of the fiber from here also have there is the contraction of this contraction ciliary muscle, and that will cause the increase in anterior curvature of lens. Okay, this one already we are knowing it. Now pupillary light reflex. Okay, it is clear up to there. Is there any confusion?
No, sir.
On pupillary light reflex, also I have also told you in the practical class that when we show the light on one eye, there is constriction of pupil on as on the opposite eye. And depending upon that, this is known as direct light reflex. Another opposite eye that is known as consensual pupillary light reflex. And adental oversponuclear that I have already discussed you that will occur in the syphilis. There will be accommodation reflex present, but pupillary reflex absent, isn't it? So that I have already discussed. Now this is the pathway, okay? Now this is the pathway, and you should be knowing why there is constriction of pupil is going on both eye or on the same side, consensual and direct. If you look at here, when the light is shown over here, now when the light is shown over here, then from here the information is going this again optic knob, and then this optic chiasma, then optic tract. Okay, optic tract. Now before, now look at it. Before going the information to the lateral geniculate body. Now look at here. There is lateral geniculate body, isn't it? There is lateral geniculate body. It is not shown here. So some of the fibers. So some of the fibers they bypass. They are not going to the lateral geniculate body, but they will bypass the lateral geniculate body, and they will synapse, or they will be terminating on pretectal nucleus. Okay, pretectal nucleus. From the pretectal nucleus, the information is going to the Edinger West Pole nucleus. Okay, parasympathetic nucleus of oculomotor. This one, and they will from same side. This is again the long one. This is short ciliary. This is parasympathetic. This one will be preganglionic fiber will be longer as compared to the postganglionic fiber. So that is pre and postganglionic fiber of parasympathetic nerve. So they will cause the constriction of pupil on both sides of the eye. Am I clear?
Yes, sir.
Anyone who is not clear, please let me know. I can repeat it again. Now, so this is the importance, okay? That now I will ask few questions to the attendee. Before that,